Okay, hi everyone. My name is Idan Frumkin, and I'm a molecular evolutionist from Tel Aviv University. I'd like to take us back to 2020, and with more than 700 million cases, that was the worst pandemic in 100 years. But this was not the first pandemic. In 2003, there was a different virus, or a different outbreak, that spread in more than 20 countries. And what's shared about these two viruses is that they come from the same original virus species. And the reason for these two outbreaks is the continuous process of evolution allowing new viral species to emerge. Meaning that the next pandemic is just around the corner, and if we want to be better prepared for it, we need to understand this evolutionary process better. My message for you today is that the birth of new genes is a key driving force in the evolution of species. Some species might be shared across many species, but some genes are really unique to species. So if we look at the fruit fly, we know, and we see in blue, that 92% of its genes are shared across different species, but 8% of the genes are unique to the fly, really creating what's unique about this creature. It's also true for humans. 2% of our genes are unique to primates and are really involved in human biology. So what do we have to gain by studying new genes? We talked about viral evolution, and new genes have been shown to allow viruses to infect new hosts. So think about a virus that can infect the bat and can now suddenly infect human and spread in its population. New genes are also involved in escaping the immune system, creating a more severe disease. New genes are also involved in cancer progression, and they have been shown to regulate tumor growth and metastasis. And finally, new genes are also involved in the bacterial life, and they have been shown to help bacteria develop resistance to antibiotics and also to create novel metabolic and regulatory pathways, really enforcing bacteria to adapt to its environment. So all of this is just to say that by studying new genes, we have a lot to gain with very strong human challenges. So how can we explain how new genes are created in evolution? There is one classic example, or a classic mechanism we know about, in which new genes come from old genes. So think about a gene or two copies of the same gene. That one copy accumulates mutations through evolution until it has completely changed its sequence, and a new gene has been created with a new function. I am interested in an alternative mechanism for gene birth that is called de novo gene birth, in which new genes come from non-functional DNA sequences. So I find that mechanism extremely exciting because it shows how evolution can create new functions from scratch, not using something that already exists, but creating something completely new. In this process, non-functional DNA can become expressed to create a nascent gene that then can be further refined by evolution to emerge until a mature new gene has emerged. In my lab, we try to think and develop new ways to study this process from non-functional DNA all the way up to mature genes. For that, we use two main sources of DNA. One is random DNA, which helps us mimic this initial stage of non-functional DNA. And the other is we are trying to find and look for mature new genes and study them to characterize their functions. In our lab, what we try to do is to use the power of microbial evolution to learn about the process of new genes, and we have three main goals. The first is to really understand the molecular mechanisms of how can new genes emerge. The second is coming from the fact that we know new genes do not emerge in a vacuum. They must be, when they emerge, they are born into a functioning, complicated molecular system in the shape of a cell, and they should perform no harm before they can do any good. So we want to understand how new genes interact with old ones. And lastly, we really want to understand the range of functions new genes can perform and allow cells to adapt with. So I want to give you one example of how we take all of these philosophical reasoning to actual experiments in the lab. In my postdoc at MIT, I created a library of 100 million random genes. Each of them has a completely random sequence that never existed in nature before. I inserted these sequences into bacterial cells. And in this specific case, I asked, can one of these random genes, or how many of these random genes, can help cells survive a virus attack? And the idea was that a virus can infect bacteria, it kills all the population, but if one or more of these random genes are able to somehow stop the virus attack and allow the bacterial cell to overcome it, then that cell will grow, we will be able to pick it and find functional random genes. And then we wanted to study how can something random that never existed before can help cells survive. From the 100 million genes, we found thousands of such cases that allow cells to stop the virus. And again, my message here is that these things never existed in nature before, so they immediately were able to integrate into the cellular system and perform something that helped them survive. They improved the cell. So with a series of experiments I will not show you today, we came up with a model of what we think is going on. Initially, the virus is able to bind the bacterial cells through what we call a receptor. It's a bacterial protein that the virus can bind and initiate the infection cycle through injection of its genome. What we have found is that the expression of these new genes that we provided somehow reduced the expression or the frequency of this receptor, hence making it more difficult for the virus to really find the bacterial cell and initiate the infection cycle. So this was very exciting for us. But what was even more fun was that we could then really mimic the ongoing bacteria-virus arms race that has been going on in nature for more than 1 billion years, and found a phage that could really counterattack this new defense by the new gene. So we found a mutate phage that was still able to bind the cell, even though it was more difficult for it. So with that, I'd like to end. My message to you is that my lab and my goal is to study the evolution and function of new genes in the hope to advance human medicine and biotechnology. For that, we use an array of classical and modern tools of synthetic biology, experimental evolution, and an array of computational tools with the one main goal of really better explaining life. Thank you very much.
Questions?